We'll now look at what happens in development during the third week, and as we'll see, the critical event that's occurring in the third week is the conversion of the two-layered embryo into a three-layered embryo. As we look at this slide, we're looking down upon the upper surface of the epiblast at the beginning of the third week of development. Now, one thing that has already happened that we can see is that the epiblast, as well as the hypoblast beneath it, has changed its shape from a circular disc into an oval-shaped structure. It has gotten longer. We see that the procaudal plate is positioned here, so we know that this is the head end and therefore this is the tail end. So it has gotten longer along what will be the long axis of the embryo. The other thing we see is that the head end of the embryo has gotten considerably larger than the tail end. So already we can see the importance of having established the polarity to know which is the long axis of the embryo and which is the head end of the embryo. We can see forming in the midline of the epiblast toward the caudal or tail end of the epiblast, we see a groove that has formed in the midline, and that groove is called the primitive groove or the primitive streak. What's happening at this primitive streak is that the cells of the epiblast from the right side of the epiblast and from the left side of the epiblast are migrating toward the midline, represented by these green arrows. So cells from here and cells from here are migrating toward the midline, and when they get to the midline where the primitive streak is, they turn and go into the plane of this image, in other words, behind the screen, and they descend toward where the hypoblast is beneath this. In order for us to appreciate that, what we will do is we'll make a section through the embryo at this level and turn it up on edge so that we can look at it, and that's what we see in this image here. Here's the epiblast. We had been looking at it from above, from this side. Here we can see the epiblast. Here's that primitive streak in the midline. And here's the hypoblast beneath it. And what we're saying is that cells from the right side and cells from the left side migrate toward the midline, reach the midline, and then descend and start to migrate in the direction of the hypoblast. Now, several different things happen to the cells that migrate in that fashion. Some of the cells migrate through the primitive streak and migrate all the way down to where the hypoblast cells are, and they invaginate that layer and start to occupy that layer. And more cells are doing that. So these epiblast cells migrate down, displace the hypoblast cells. The hypoblast cells get pushed aside and then will eventually die. They're replaced by these epiblast cells that are now occupying that layer, and we now call those cells endoderm. So endoderm cells are cells that were epiblast cells, migrated through the primitive streak, and are occupying a position that used to be occupied by the hypoblast. Other cells that migrate through the primitive streak migrate toward the hypoblast but don't go quite as far, and they will end up entering the space between the two layers. Recall, we said the epiblast and hypoblast are separable from one another, so we can create a space between them, and these cells are then occupying that space in a position such as this, and more of those cells end up occupying that space, and those cells that are now in the middle layer between the epiblast and what used to be the hypoblast, those cells that are in the middle, since the prefix for middle is meso, we call those cells mesoderm. So those are the mesoderm cells. And you notice that these epiblast cells that become mesoblast, when they migrate through the primitive streak to enter that middle layer, they then either migrate toward the right side or migrate toward the left side of the embryo. There are other cells from the epiblast that also migrate through the primitive streak, also become part of the middle layer, but those cells stay right in the midline of the middle layer, and those cells are then called the notochord. Lastly, we have cells of the epiblast that don't migrate through the primitive streak. They remain in this upper layer, or the epiblast. And those cells that remain in the epiblast layer while the other cells have migrated through the primitive streak, those cells change their name and are now called the ectoderm. So we now have the establishment of a three-layered embryo. The upper layer is ectoderm, the middle layer is mesoderm and notochord, and the lower layer is endoderm. But remember, all of the cells from all of those layers were originally part of the epiblast, and that's why we can say that it is the epiblast that gives rise to all of the cells of the future embryo. The cells that remain in the epiblast and are now termed ectoderm will adopt different names depending upon which part of this upper layer they occupy. Those that occupy the upper layer near the midline will become called neural ectoderm cells. Cells that are right alongside of that will be called neural crest cells, and the cells in the remainder of the upper layer will become either skin or surface ectoderm cells. So the ectoderm will get subdivided into three kinds of ectoderm. The middle layer is mesoderm and notochord. The lower layer is endoderm. We see this chart with three columns, with the three columns representing the three layers. So we see one column for ectoderm, one column for mesoderm, one column for endoderm. And beneath that, on this slide as well as on the subsequent slides, we see adult structures that are the derivatives of those cell types. So we see that there are things that come from ectoderm, things that come from mesoderm, things that come from endoderm. Now, at this stage of our discussion, we don't want to be just memorizing the names of these structures because that's not a way that we'll learn them. What we will be doing, in fact, over the course of our continuing discussion is be talking about each of these structures in their appropriate place, and when we do, we'll go back and understand how those structures came from those layers. There are just a couple of general points that I'd like to make while we're looking at this slide. The cells that are derived from the endoderm and the cells that are derived from the ectoderm all end up being epithelial cells or epithelial-derived cells. So, for example, under endoderm, we see that it says it forms the epithelial lining of the GI tract, and it forms the epithelial lining of the respiratory system, and it forms the epithelial lining of some parts of the urinary system, for example. So it's not forming all of the GI tract or all of the respiratory system, but rather only the epithelial lining of those systems. Similarly, the ectoderm is also forming epithelium, and that's why you see, even though we will call the surface or skin ectoderm, it doesn't really give rise to skin. It gives rise only to the epidermis of the skin, not the dermis, because it's only the epidermis that's epithelial. The dermis, which is connective tissue, is going to be, in fact, derived from the mesoderm. And all of these other structures that we see are all epithelial-derived cells. The mesoderm, on the other hand, gives rise to many different cell types, and we can see it gives rise to muscle and bone and cartilage and internal organs and blood vessels and so forth. We also said that the middle layer includes the notochord, and the notochord gives rise to only one structure in the adult, and that structure is the nucleus pulposus of the intervertebral disc, which we'll talk about later. So even though the notochord seems to have a very limited usefulness, it gives rise to only this one adult structure. In fact, the importance of the notochord is that it's sort of a director of what's happening in the embryo. Because of its central position, during development, the notochord is releasing substances into its surrounding area to direct the cells around it to differentiate in different ways. So it's the major organizer of embryonic development. Once it has finished doing all of that, the only adult structure it continues to become is the nucleus pulposus. We said that part of the ectoderm is going to become neural crest ectoderm, and that will give rise to a variety of structures, which we'll talk about later on. And part of it will be neural ectoderm, and that will give rise to a variety of structures that will be talked about primarily with the neuroscience portion of this review program. So we now have a three-layered embryo, and we have the cells that will give rise to all of the cells of the adult. But the one problem we still have to solve is that what we have is a flat three-layered embryo. If we can go back to our model here, I've converted our two-layered embryo of epiblast and hypoblast into a three-layered embryo by creating a middle layer. So we now have three layers: ectoderm on the top, mesoderm and notochord in the middle, and endoderm on the bottom. But it's flat. And the adult body structure is not flat, it's actually round. So to convert this three-layered flat embryo into the adult structure, what we need to do is convert it into a round three-layered embryo. And all we need to do to accomplish that is to have it fold. And this is what occurs in the fourth week. We go from this flat three-layered embryo, and simply by folding it around like this and having the edge on the right and the edge on the left meet and fuse in the midline, we've now converted this from a flat three-layered embryo into three tubes: an outer tube of ectoderm, an inner tube of endoderm, and in between those two, a layer of mesoderm. We see that the tube is open at the front end and it's opened at the back end. So this inner layer of endoderm, which we already said will become the lining of the GI system, we therefore can say that this will become the lining of the gut tube. It's open at the front end, that's where the mouth will be, and it's opened at the back end, that's where the anus will be. And so we have a tube that runs all the way through the embryo, completely lined by endoderm. We have the outer surface completely lined by ectoderm. We said that the skin, the epidermis of the skin, is going to be coming from ectoderm. And then everything in between is going to be mesoderm. And we said that the mesoderm gives rise to cartilage and bone and muscle and connective tissue and so forth. That, in fact, is what we find between the surface of the body and the lining of the gut tube is going to be connective tissue and muscle and bone and cartilage, and that's all coming from this middle layer of mesoderm. So by converting this flat three-layered embryo into a round three-layered embryo, we've established the adult body form. And we can see on this slide a representation of how that is occurring. If we just look at a part of this, here's the layer of ectoderm, here's the layer of endoderm, here's the mesoderm in between. We'll ignore this and ignore this for now. And what we see happening is this three-layered structure is starting to fold around, with this edge, which was over here and here, now coming closer to one another. The endoderm layer is starting to become a tube. Some of it is left outside of the embryo. And then finally, this surface and this surface will fuse, causing this tube to be internalized. And we now have a tube inside a tube inside a tube. The outer tube is ectoderm. The inner tube is lined by endoderm. And we have mesoderm in between. That will become the lining of the tube. That will become the surface ectoderm, forming the epidermis of the skin. But recall that in order for this to occur properly, that edge and that edge has to fuse in the midline to close off this ventral surface. And normally that does happen. But one of the generalities we can make about embryonic development is that when cell layers are supposed to fuse, sometimes they don't fuse perfectly. And when there are imperfections in the fusion, then we're going to end up with various congenital defects. In this case, if there's an imperfection in the fusion of the two lateral body folds, we will have a gap in the ventral body wall. And that gap in the ventral body wall will allow structures that should be inside the embryo to escape outside of the embryo and end up out here in the amniotic cavity. That kind of a defect is given the name gastroschisis. So gastroschisis is a result of a defect in the fusion of the lateral body folds, resulting in a ventral body wall gap or defect, which will allow abdominal viscera to escape from the abdominal cavity into the amniotic cavity. And those organs will then be in contact with the amniotic fluid in that amniotic cavity. So that brings us to a conclusion of the first 4 weeks of development. We have the basic body plan established, and what we will now do as we proceed forward is look at adult anatomy in one region after another, and as we get to each major organ system, we'll come back to this 4-week-old embryo and take some small of that 4-week-old embryo and develop it into the adult. So that when we're talking about the adult heart, we'll talk about the development of the heart. The adult GI tract, we'll talk about the development of the GI tract, and so on.